All right, so now we are to the next segment here of the reproductive system. We're going to finish up the male reproductive system, and then I will talk just kind of beginning parts of the female side of things, but again, a little bit more complicated in terms of the hormones for the female side. So we're going to kind of work our way through it. It's going to take a little bit longer, so I'm going to finish it up in the next and last segment, probably of this chapter. Okay, so we were finishing talking about the differences between meiosis and mitosis and kind of how the gametes are formed and how they get to that haploid number of chromosomes. Now, the sperm production really takes on full effect at puberty. Of course, a lot of changes happen at puberty, and that's one of them. The spermatogonia, these are the stem cells that are in kind of what we say, the basal compartment of the seminiferous tubule, the side that is the closest or the furthest away from the lumen of that tube, okay, the most superficial part really of those tubes. And they undergo mitosis, and each mitotic division of those stem cells give rise to what we call a type A daughter cell and a type B daughter cell. The type A stays behind and acts as another stem cell to give rise to, again, two more cells. They'll undergo mitosis again and again. The type B are the ones that are selected to go further into the wall toward the lumen of the seminiferous tubule and develop into those primary spermatocytes. And so from spermatocytes to spermatids, we undergo meiosis. So in meiosis 1, we have the primary spermatocyte, which is still 2N. It divides and gives two secondary spermatocytes that are each haploid. And then in meiosis 2, you have those two secondary spermatocytes that undergo really basically a mitotic event, but we call it meiosis 2, and those develop into two spermatids. Now again, spermatids are not fully functional yet. They're not fully mature. And so they're small, they're non-motile, but they are really close to the lumen of the seminiferous tubule. So if you think this is the outside wall of the seminiferous tubule up here, these are the stem cells. They divide, it gives rise to type A that stays behind and type B that moves toward the lumen of the seminiferous tubule. So it undergoes meiosis 1, it creates these two secondary spermatocytes. Those then undergo meiosis 2, creating 4 spermatids. And again, they're not fully functional yet, they still have some development to do, but they're making their way all the way to the lumen of the seminiferous tubule. So now, how do we get from that kind of immature version of a sperm called a spermatid all the way to fully functional sperm? And that is the process called spermiogenesis. And so the spermatids have the right number of chromosomes. We split it all up, we're good to go there. But there's still some changes that have to be made. They're still not motile, so we've got to give them motility. And so through the process of spermiogenesis, the spermatids elongate, they lose excess cytoplasm in the cap region, and they form a tail. And then finally, once they have that, then they're full-on, full-fledged sperm at that point. And so you can kind of see this process take place. So this is a spermatid over here. Again, they're losing excess cytoplasm. That's getting less and less. They're developing this tail. And then finally, you have this fully functional, fully developed sperm by the end of it. So the major regions of the sperm are the head, which is really where the nucleus is, where the chromosomes are. It has a little acrosome cap at the top of it that contains enzymes that are going to be able to eat away at the wall of the ovum to be able to get that genetic material into the interior part of the egg. The midpiece is what we call the metabolic region. There's a bunch of mitochondria here that their job is to pump out a whole lot of ATP so that we can have mobility. And then, of course, the tail is what gives the sperm its mobility. It's the locomotor region. Now, in the tube, or in the seminiferous tubule, the walls themselves are made up of cells called sustentacular cells. There's actually lots of names for these cells. Sustentacular cells, sustenocytes, I think is another name, nurse cells or Sertoli cells. Those are all names for the same cell, as confusing as that is. But they make up the walls of the seminiferous tubules, and they surround the developing spermatids. So their job is to provide nutrients, like they're nursing these developing spermatids. We think of it that way. So they provide nutrients and signals to those dividing cells, and they help those cells move toward the lumen as they develop. They also secrete the testicular fluid that is in the lumen that's going to help with sperm transport. And if there's any kind of wrong division that happens, or maybe too much cytoplasm, they also get rid of that as well. They get rid of non-functional cells. They just kind of clean up the area. And they produce chemical mediators to regulate spermatogenesis. And we'll talk more about that in just a little bit. There's also tight junctions that exist between adjacent sustentacular cells, which helps to divide the seminiferous tubule wall into two components: a basal compartment where the spermatogonia and primary spermatocytes live, and then the adluminal compartment, which is deeper to the basal compartment, where the meiotically active spermatocytes are, right? So it's more toward the lumen. So if you look at this picture, again, we have the spermatozoan spermatocytes going through, developing all the while, right? But these yellow cells, this kind of yellow area, are the sustentacular cells, right? So they have their own nucleus here, okay? And there's tight junctions right here, represented here, that separate into this basal compartment and the adluminal compartment, right? There's going to be another really important job of these little tight junctions here in just a little bit we'll talk about again, which is actually, we'll talk about it right now. So they help to make what we call the blood-testis barrier. We all know about the blood-brain barrier and kind of its job. This is similar, although not as, it's a little bit more penetrable, I guess you could say, than the blood-brain barrier. But their job is to prevent sperm antigens from getting into the bloodstream of that male, and therefore activating the immune system. And if you think about it, our immune system is developed pretty early in life. You know, the adaptive side of our immune system, right? Once we're exposed to different antigens, it develops against those. And by the time puberty hits, we have a pretty fully functional immune system. Well, these sperm aren't made until after the development of the immune system. So technically, they are viewed to the male immune system, they are viewed as non-self. Even though they are technically self, they are viewed as non-self because they were made after the full development of the immune system. So this blood-testis barrier protects the production of sperm, so they're not being killed by the immune system of that male, right? That's why that's there. Now, this whole process takes anywhere from 64 to 72 days if everything is nice and hospitable. The pressure of this testicular fluid from those sustentacular cells pushes those non-motile sperm into the epididymis, where they get their full motility and fertilizing power. And a lot of this is all driven, of course, by hormones. There's a sequence of hormone events that involve the hypothalamus, the anterior pituitary, and the testes. So there's an axis here involving those three. We have what we call the hypothalamic pituitary gonadal axis, or HPG. It's going to be similar, but with different hormones in the female reproductive system. So this helps to regulate the production of gametes and the sex hormones through a set of three sets of interacting hormones. So first we have GnRH. GnRH is gonadotropin releasing hormone. If you think back to the endocrine system, which I know seems like forever ago. 2020 has been a long year, a long decade, 2020. But that's what we talked about at the very beginning of the semester. And so it directly stimulates the testes, indirectly stimulates the testes via luteinizing hormone and follicle stimulating hormone that come in from the anterior pituitary. Those two hormones talk directly to the testicle and tell it to make hormones. And then one of those hormones that is made is, of course, testosterone. And inhibin both, they feed back and turn off the HPG axis. So there's not just an overloading amount of these hormones around all the time. So here's kind of the sequence of the events. The hypothalamus releases GnRH, and that tells the anterior pituitary to release FSH and LH. FSH's job is to talk to those sustentacular sites and tell it to secrete a protein called androgen binding protein. And what this does is it binds androgen and keeps a really high concentration of it near the developing sperm. And the androgen that we care about is testosterone, right? Luteinizing hormone prods the interstitial endocrine cells of the testis, which are kind of outside the seminiferous tubules. We haven't really talked about those yet. They're outside the tube itself, but they produce testosterone. And of course, that prods spermatogenesis. Now, testosterone does a whole bunch of other things too. You know, sex organ maturation, development of the secondary sex characteristics, libido is triggered by testosterone. As testosterone levels rise, it negatively feeds back to the hypothalamus and tells it to reduce production of GnRH, and therefore not necessarily turning off this entire axis, but turning it down to manageable levels. Inhibin also is a hormone that's released from the testis when sperm count is high. And so it's going to feed back and again inhibit GnRH and FSH release. Now, once puberty hits, and we all know puberty is one of the most awkward times of our life, right? It takes about 3 years for balance to be achieved. And, you know, you've got the voice changes that are happening. You have a lot of characteristics, secondary sex characteristics developing. Hair is starting to grow, you know, different things that are happening. But it takes about 3 years to achieve balance. And then in a male reproductive system anyway, testosterone stays high and it just stays up there really throughout the rest of that person's life until very late, you know, late decades, like late 80s, 90s, then you start to see some testosterone decline. But if there's something wrong with that axis, maybe there's some trauma to the hypothalamus or the pituitary, if there's no GnRH and there's no gonadotropins, then the testes start to atrophy. Sperm and testosterone production will cease, and you know, just have no reproductive power at that point. All right, so here's this axis. Here's the hypothalamus up at the top, secreting GnRH. GnRH tells the anterior pituitary to release follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone secretes the androgen-binding protein, which concentrates androgen in this area, in promoting spermatogenesis. Luteinizing hormone tells these interstitial cells, again, that are outside the seminiferous tubules, but inside the testis still, to secrete testosterone, which also prods and stimulates spermatogenesis. But again, this testosterone will feed back and kind of turn off this axis, as well as inhibin that comes from the testis will feed back and turn off, or at least turn down this axis. All right, so mechanisms and effects of testosterone activity. Testosterone is a steroid hormone, lipid, that's synthesized from cholesterol, and it exerts its effects on some of the target cells that, like, it turns into dihydrotestosterone in the prostate, or estradiol in some of the neurons in the brain. But its main job is really to prompt and start and keep going spermatogenesis. And it also targets all of the accessory organs of the male reproductive system. So there's a lot of anabolic effects throughout the body that testosterone causes. One of the things we talked about earlier in the semester was how it triggers more red blood cell production. It's typically why the hematocrit in males is more or higher than in females, is because of testosterone. Deficiency leads to atrophy of the testicles. Semen volume declines, erection and ejaculation is impaired. Usually this is treated with testosterone replacement therapy, but with that, sometimes will lead to reduction in sperm count, because again, you have that inhibition of the HPG axis. So some of the secondary sex characteristics, kind of features that are in our non-reproductive organs, are appearance of pubic, axillary hair, and facial hair. Enhancement of growth of the chest hair, deepening of the voice, the skin thickens and becomes more oily, bones grow and become more dense, muscles also grow and increase in size and mass, metabolic rate gets higher. It's one of the reasons why if men just have to think about losing weight, and they do, it's because testosterone is so metabolically active, and that causes their metabolic rate to be higher. The sex drive comes from testosterone as well. And so there's a lot of things that testosterone does besides just make sperm that are important to overall reproduction, really. So testosterone also masculinizes the embryological brain, and it continues to exert its effect well into adulthood. The adrenal glands also secrete a little bit of androgens. Remember the 3 zones of the cortex of the adrenal gland, right? One of them was gonadotropins, is what it released, and that can then turn into testosterone. It's not enough to maintain normal effects of testosterone, but it can help in that. All right, so if you're looking at kind of age here, in terms of when testosterone is secreted, there's a spike during the embryological stage of life. And what happens here is that at the beginning of, like right after conception, yes, genetically, there's a difference, you know, XX with female, XY with male, but the embryo looks very much the same. But there's finally a pathway that it takes at around 3 or 4 months or so after embryological age. And that's where you're going to start to see the spike either in estrogen or or testosterone that is going to prod the development of the different sexual organs, right? It's going to really split that pathway. So that's why you see the spike here. And then, of course, by puberty, you see this big production rise in testosterone that is also subsequently followed by a production of sperm, right? And then again, that continues well throughout life, stays high. There's no drops. There's no monthly change like we'll see with the female reproductive system. And that just keeps sperm levels high throughout life. And again, there's a drop later on in life, but it's really, really much later. Okay, so that's really it for the the male reproductive tract and system. We're really going to now concentrate more on the female system. I'm going to kind of introduce a little bit of this one, and then we'll continue it on in the next segment. So for the anatomy side, of course, the gametes or the gonads are the ovaries, which produce the ova, or the gametes for the female. It also secretes some of the hormones that are important, estrogen, different types of estrogens, progesterone. And some of the accessory ducts are the uterine tubes, the uterus, the vagina. The internal genitalia are inside the pelvic cavity, and really that's most everything. There are a few external genitalia structures, but most all of the female reproductive system is inside the pelvic cavity. You can kind of see that here. Here's the ovaries. This is the oviduct or uterine tube, the uterus with the cervix, and then the vagina, and then these would be the external genitalia out here. Now, physiology-wise, it's kind of always been assumed that male or females were born with their determined amount of eggs, and that there's no way to make more eggs at that point. There's been a lot of new evidence to suggest that some stem cells can give rise to more cells at the ovary surface, but I don't know how new this information is. I haven't looked to see if this has changed, if the dogma behind this has changed, where now we know that we can make more eggs, or if it's just, it might still be the case and they're not 100% sure. We're going to go with the assumption that females are born with a set amount of eggs and just kind of work our way through the chapter with that for now. But just know that there may be some discoveries that are happening that may overturn that assumption. Okay, so let's talk oogenesis. Now, the production of gametes in the female is very similar to the production of gametes in the male. There are some differences, but if you still have meiosis 1, you still have meiosis 2, you're still going to go from diploid to haploid. All of that is still going to happen. Some of the differences are it takes a lot longer for oogenesis to complete. It begins in the fetal period of life. It stalls and then gets taken back up at puberty, at least 1 or 2 ovum every month. And then it even stalls before conception. And the only time it really ever finishes is if conception happens. Right? We'll see that as we go along. So oogonia, those are the stem cells, the 2 end cells that give rise to the developing eggs that, they undergo mitosis, and it's kind of like the type A and the type B that we saw in the sperm, what we're dealing with here. So it'll give rise to a primary oocyte that will develop from those primordial follicles. And those primary oocytes develop or begin the meiosis event, and they stall in prophase 1. So they hang out there for years, right? So at birth, again, we're going to presume that we have a lifetime supply of primary oocytes, and then after puberty hits, a few of those primary oocytes that are stalled in prophase 1 resume, right? One is selected and it resumes, and it creates 2 haploid secondary oocytes at the very end of that. It's a large cell with all the cytoplasm and organelles that a typical cell will have. And then the secondary, or the second cell that comes off of that meiosis event is called a polar body. This is a very tiny cell. It is not going to develop any further than that. We'll see what that looks like here in just a second, right? But that's one of the differences that I want to point out, is that these oocytes, when they're developing, we're not going to give rise from one germ cell, one stem cell. It's not going to give rise to 4 equal ova, like we saw with the sperm. we're only going to get one functional ova. Okay. The first split here, we have the ovum, the secondary oocyte, and then this tiny little, basically fragment of a cell called a polar body. All right, so that secondary oocyte is going to rest in metaphase 2, and that's what's ovulated from the ovary, goes into the fallopian tube, and then should conception occur, should a sperm enter into that ovum, then it'll complete meiosis. If it doesn't, it just deteriorates. It never completes it, right? So if it completes meiosis 2, it yields a functional gamete, the ovum, and the second polar body. All right, so let's look at this here, this left side of this picture. Before birth, again, those little germ stem cells, the oogonia, are going to undergo mitosis and create the primary oocyte. Those primary oocytes are going to go into meiosis 1, but stall in prophase 1. And then finally in puberty, one of them is going to pick back up, continue into meiosis, and finally it'll finish up meiosis 1. It'll create the secondary oocyte here and the little first polar body. That polar body is going to go on to meiosis 2 and split just like normal, but it's going to create 2 polar bodies that we really don't care anything about. They're going to deteriorate anyway. But here, the secondary oocyte is going to start in meiosis 2. It's going to stall in metaphase 2, be ovulated, and should a sperm penetrate it and join in, should conception happen, it'll finally finish meiosis 2 and create this fully functional ovum, right? And that usually happens in the fallopian tube or the oviduct. And then you also have the second polar body. So at the end of a meiosis event for an egg, you do not have 4 functional eggs. You have 1 functional egg and 3 polar bodies that just go off and get deteriorated, right? Whereas in the sperm, you have 1 to 4 fully functional sperm, should everything go right, of course. Now you'll notice too on this side on the right, you'll see that the size of the oocyte or the follicle gets larger and larger and larger as it matures. And then during ovulation, we're only ovulating this like kind of interior part of the tertiary or Graafian follicle, and that moves into the fallopian tube. We'll talk more about that later. So again, kind of comparing oogenesis and spermatogenesis. Spermatogenesis gives rise to 4 viable sperm. Wow, can't talk this morning. But there's an error rate of about 3 to 4%, which really is pretty good. I mean, you think about how many sperm are developed, which is a lot. An error rate that small is pretty good, right? In oogenesis, you only get 1 viable gamete from every 1 germ cell, stem cell, and 3 polar bodies. And it has an error rate of 20%. So not only are we not producing as many gametes, but we're also producing them at a higher error rate, okay? This unequal division that we see in oogenesis really ensures that the developing oocyte inside the follicle has enough nutrients and enough organelles and all of that to complete what it's going to have as a basically 7-day journey onto the uterus. The polar bodies just degenerate and die, right? So that's where we're going to stop for now. We're going to pick up in this last segment. We're going to talk about the ovarian cycle, the monthly cycles that happen at the ovary. We'll talk about the uterine cycle. They do go hand in hand together, but we do kind of talk about them separately because there's different events that are happening in each of those. So we'll pick up there in this next segment, and I'll see y'all then.